All right, let's dive into the world of biology. We're going to go beyond just the big, cool ideas about life itself and really get into the nitty-gritty, the essential rules that make discovering new things not only possible, but safe. Let's get to it. So here's the game plan. First up, we'll tackle the big picture. What even is biology? Then we're stepping into the lab to learn the crucial rules of engagement and, how not to blow things up. From there, we'll see how scientists actually talk to each other through their work. And finally, we'll put it all together to learn how to think like a scientist. So what exactly are we talking about when we say biology? Look, it's so much more than just a subject you take in school. When you get right down to it, biology is the story of every single living thing on this planet. And yeah, that includes us. The name itself kind of gives the whole game away. It comes from two Greek words, bios, which means life, and logos, which means study. So biology is quite literally the study of life, how living things work, how they get along, and how they fit into this big, complicated world. And when I say the study of life, I mean it is massive. It branches out into all these amazing fields. You've got botany, which is all about plants, ecology, which looks at how we all interact with each other, physiology, which is the nuts and bolts of how bodies work, genetics, the literal code of life, and even microbiology, which explores that whole invisible world of tiny organisms all around us. And the impact of all this study? Oh, it's absolutely huge. It's in the food we grow, thanks to things like transgenic crops that can fight off diseases. And it's in medicine, with mind-blowing stuff like gene therapy that could one day cure diseases we inherit. Biology isn't just in a textbook, it is actively changing our world. So where does all this incredible discovery actually happen? Well, most of the time it's in the lab. Think of it as the arena, where ideas get put to the test. But it's also a place where you have to balance that curiosity with a healthy dose of caution. So let's talk about the rules of engagement. Okay, first things first. Before you even think about starting an experiment, you gear up with your personal protective equipment, or PPE. And this isn't about fashion, it's about staying in one piece. Your lab coat is your armor against chemical splashes. Goggles are non-negotiable for protecting your eyes. Gloves are for when you're handling sensitive samples. A mask keeps nasty fumes out of your lungs. And proper shoes, well, they protect your feet from broken glass and spills. This is your essential toolkit. Now, every piece of equipment in a lab is basically a solution to a problem. For example, some chemicals give off invisible vapors that are seriously dangerous. Think chlorine gas. You can't see it, but it's there. So, what's your defense? The fume hood. It acts like a super-powered vacuum, pulling all those hazardous gases safely away from you. But what if the worst happens and a chemical actually splashes on your skin or in your eyes? That's a serious hazard. Your immediate defense is the emergency shower and eye wash station. You don't hesitate. These are designed to drench you with water instantly to rinse away anything dangerous. All right, pop quiz. Let's say a thermometer breaks. Bam, now you've got a mercury spill on the floor. What do? I'll give you a hint. You do not grab a paper towel and wipe it up. That's a big no-no. There's a very strict protocol for a reason. Step 1, tell your teacher. Right away, don't touch a thing. Step 2, make sure everyone knows to stay clear. That area is now a restricted zone. Step 3, you sprinkle sulfur powder over the spill. This reacts with the mercury and helps contain its toxic vapors. And step 4, you call in the pros, the fire and rescue department. Following these steps isn't just a suggestion, it's absolutely critical. Now, a huge part of being safe in a lab is knowing what to do with stuff when you're done with it. It's all about mastering biological waste, because in a lab, you can't just throw things in the trash. Every single material has its proper place. Let's start with something you might think is simple. The sink. Nope. There are hard and fast rules. Things that are pretty harmless, like dilute sugar solutions or stuff with a neutral pH, are usually okay to go down the drain. But things like organic solvents, heavy metals like that mercury we just talked about, any solid waste or really strong acids and bases, absolutely not. Never. They need to be handled separately. You see, the whole system is super organized, broken down into categories. Category A is for sharps, syringes, needles. They go straight into a special sharps bin, no questions asked. Category B is for non-sharps, like used gloves or petri dishes. Those get packed in a biohazard bag and sterilized in a machine called an autoclave before they're tossed. Category C, carcasses or tissues, get wrapped, bagged, and frozen. And finally, Category D, liquids, like blood, have to be decontaminated, usually by autoclaving, before they're disposed of. Every category has its own strict protocol. Finishing an experiment safely is really only half the battle. Now you have to show people what you found. And to do that, science has developed a kind of universal language, a set of tools that make sure your work can be understood by any other scientist anywhere in the world.
Let's look at a classic example, bar charts and histograms. They look pretty similar, right? But they tell totally different stories. A bar chart, with those gaps between the bars, is for comparing separate things, like say the number of apples versus oranges sold. A histogram, where the bars are all touching, shows you how data is spread out over a continuous range, like how many students fall into different height ranges. Using the right one is crucial for telling the right story.
And then there's drawing. Listen up, because a biological drawing is not a piece of art. It's a piece of data, and the rules are non-negotiable. You use a sharp pencil and only a pencil. The drawing needs to be big and accurate. Your lines have to be clean and solid, no sketchy fuzzy lines. Absolutely no shading or coloring. You use a ruler to draw straight lines for your labels, and those lines can't cross each other. And finally, you always state the magnification. Precision is everything.
So we've got the safety rules for the lab, and we've got the language to communicate our findings. But what's the glue that holds it all together? It's the mindset. It's about learning to think like a scientist, and there's a method to that madness.
And here it is, the scientific method. This is the blueprint that turns a simple question into a real discovery. You start with a problem you can actually test. Then you make an educated guess. That's your hypothesis. You carefully plan your experiment, figuring out your variables. Then you do the work, you collect the data, and you analyze it to see what it's telling you. From that, you draw a conclusion. And the final step, you write it all up in a report so other people can learn from what you did.
That planning stage is so important, and it's all about understanding your variables. The manipulated variable is the one thing you're intentionally changing to see what happens. For example, the amount of exercise someone does. The responding variable is what you measure to see the effect. In this case, you would measure their pulse rate. And the fixed variables? Those are all the things you have to keep exactly the same to make sure it's a fair test, like using people of the same age and fitness level.
And underneath all of this, the safety gear, the disposal methods, the charts, the scientific method, there is one core value that holds it all together: integrity. A scientist has to be honest and unbelievably accurate. The data has to be real. It has to be validated, because without that foundation of trust, the whole thing just falls apart.
But where does any of this even start? Every experiment, every major breakthrough, every single scientific paper ever written, it all started with just one thing: a question. It all begins when someone gets curious enough to ask why, or how, or what if.
So let's bring it all home. You now have the fundamental toolkit of a biologist. You have the rules to explore your curiosity with caution. You have the language to share what you find with the world. And you have the method to guide your journey from a simple question to a solid conclusion. The only real question left is, what will you discover?